So in this part of the lecture, I will summarize the results of our analysis where we compared two groups and we used all the streamline data for all the disks and all the bundles. And as I mentioned, these results were published in the BUAN paper. So here, how can we do that? Well, here I'm presenting a summarized result where for each disk, we're testing if the two groups are different. So we're testing if this difference, the estimated difference, beta diff, is significantly different from 0. So here you can see that the sum of the p-values, and we're plotting that on the minus log 10 scale. So 2 corresponds to a p-value of 0.01. So for some of the disks, for one of the tracts, actually there seem to be differences between the two groups. Now, we have done this analysis for many tracts, for both left and right tracts, and here we are presenting results for the fractional anisotropy when we're comparing controls and patients. Here, those horizontal lines are actually at the level of 2 and 3, corresponding to the p-values of 0.01 and 0.001, or 10 to the minus second and 10 to the minus third. You can see here in row 3, column 1, there's one of the tracks where actually the differences are quite significant because the p-values reach 10 to the minus 5th here. Okay, now, this is all nice, but it would be even nicer if we could present those results back on the tracks themselves. So here, let me concentrate on the top plot here, and I'll be going through kind of A, B, C, and D subplots. So in plot A, you can see the tract here with the disks that are presented or that are in different colors. So now along those disks, you can see in figure B, we're actually analyzing data for different measures for each of those disks. So here, I know these are pretty small figures here, so you might not see really the labels. Everything is nicely printed in the paper, and this is a plot from the paper. In the top left corner, in this panel of 4, we have FA values, fractional anisotropy values. Here in the top right, we have values from the mean diffusivity, and we have 2 other kind of comparisons here in the bottom plots. Now, after we do such an analysis, we can go back to our bundle in figure C, and we can actually show the differences for the disks where those differences are present. So here we found that the differences are present for disks 58, 59, 60, 61, and 62. So here we show it in yellow portion on otherwise green tract, significant differences. Then for those disks from 58 to 62, we can extract actually values, for example, for FA, MD, and this is presented in panel D, where we are looking at the detailed values for all those disks where the significant differences were found. So here what we can see is that there are differences between patients and controls in the panel D. Now, what are the issues that we haven't addressed yet? We're analyzing the data across 100 disks, across many bundles. So we have to deal with the multiple comparison problem, how to kind of adjust for multiple comparisons that we are making. Right now, we're leaving that to the user, but in the next version, we'll be actually implementing ways that take into account the correlations between the disks. So we're not adjusting like purely, but let's say Bonferroni correction, which would really underpower our studies. Also, so far we haven't adjusted for possible confounders, but that's easy within the framework of the linear mixed models. So this multiple comparison and this correlation between disks, I would say these are more important problems that need to be taken into account. So to conclude, what have we done here? Well, data. And when I say data, this might be fractional isotropy, this might be mean diffusivity, this might be different, more complex measures that can be obtained from the diffusion-weighted imaging. And we extracted all of that along streamlines. Now, I call it registration, but it's actually an alignment, or you can think about trying to accumulate data from all the streamlines within a bundle, so the comparisons can be truly made. Unless we do something like that, it's very hard to make any statistical comparisons across groups of people or groups of observations. Now, how we analyze the data, we analyze this data via a hierarchical model. You can think about linear mixed model as one of the examples of that, where we might have correlations at different levels. We might have correlations within streamlines, within tracts, within subjects. So the open problems that we are still working on is this incorporation of spatial proximity. So data from disks that are close to each other, this data are correlated, we have to take that into account. And as I mentioned, one of the approaches that we're working on is this approach of functional data analysis that we're in the middle of finishing that project. And the last thing is that actually we have shapes that actually were discussed by Bramshandio in the earlier presentation. So this is also something that we want to take into account in the analysis. So thank you for your attention and hope you enjoyed this talk and all the other talks during the workshop.